Lucy. And this is Maria. They're both very pretty, no? Well, what if I told you that they're actually twins? That's right. Lucy and Maria Eilmer have the same birthdays and parents, but not all people believe them. Now, while the odds of this happening are very small, something smaller can be looked into to help us predict when and explain why something like this occurs. In order to understand better, we'll be doing a series of activities you can do too. Before we commence any activity, this sound will play in order to inform us when we need to gather our materials. The reason why we're doing these experiments is, again, to help us understand better, because we're getting hands-on and we actually get to see, the concepts as they play out right in front of us. For now, think of a group of people, i.e. a population, as a gene pool where we will look deep inside them to determine the allele frequency. For a quick recap, each trait, whether genotype or phenotype, has two alleles, and a single allele can either be dominant or recessive. Putting two alleles together can either make them homozygous or heterozygous, depending on what kind of combination was made. Fun fact, when you catch a virus that you've never had before and you get better from it, that's an example of microevolution. Still, with what's happening now, better be safe than sorry. The Hardy-Weinberg equilibrium is what we'll use to generally determine the percentage of a population being homozygous recessive, homozygous dominant, or heterozygous. Variation is natural, but according to this principle, the amount of variation would remain constant from one generation to the next as long as a certain criterion is met. 1. No selection. 2. No mutation. And 3. Large population. Our first activity! To test this and all the other activities for later, you'll need 100 pieces of 1-inch squared white paper, which will represent single recessive alleles, 100 pieces of 1-inch squared colored paper, which will represent single dominant alleles, a small container, and 2 small paper or opaque plastic bags. For our first activity, we tested out the Hardy-Weinberg equilibrium after generations of sexual reproduction. Step 1. We combined 50 pieces of the white paper and 50 of the colored into a plastic bag, then noted that our starting frequency of the alleles was 50 by 50. Number 2. Without looking, we drew 2 pieces of paper and took note of its genotype. Number 3. We returned the papers to the bag and Hanny, that bag was shook. Number 4. We repeated the process about 50 times, remembering to note the genotypes we picked out. Number 5. We counted the number of times we got each genotype, then calculated the frequency. Number 6. Based on the 50 individuals that we picked out, we calculated the new allele frequency using this formula. And here, somewhere, are our results. Our results show that in this population, it can be concluded that the allele frequency was ever so slightly affected by sexual reproduction in the direction of the recessive trait by 0.1%. If someone asks you how many diploid individuals you got, don't be confused, it's 50. Since we basically picked out human individuals, they're all diploids because they have 2 alleles, each one inherited from a parent. Evolution is basically a change in the traits of a population that are able to be passed on over generations. Lots think that natural selection and evolution are very synonymous, but here's another fun fact. Natural selection is only one of the mechanisms of evolution, and it is certainly the most popular. So let's shine the spotlight on another mechanism. Genetic drift. The main difference between natural selection and genetic drift is that the latter comes at random, meaning that no one has an advantage in surviving or reproducing. Because of that, it's hard to keep track of what kind of allele frequency would be the outcome. Honestly, I thought it was a bit confusing, but that's okay. That's what the activity is for. Number 1. One of the bags became the gene pool of interest, while the other became the reserve bag. Number 2. We pulled out one individual or two papers like in the previous activity, then recorded its genotype, remembering not to put it back in the bag just yet. Number 3. We repeated for a total of 5 times, which equated to 1 generation. Number 4. Afterwards, we computed the allele frequencies for this generation. Number 5. We then adjusted the allele frequency in the bag to match the allele frequency we just calculated for this generation. Finally, we repeated this for 5 generations, each time calculating our allele frequencies and then adjusting the bag. This is what we came up with. Here, for the second activity, the allele frequency changed from generation to generation in the direction of the recessive trait. Because the genetic drift concept states that there is no clear way to predict the allele frequency due to it being centered on chance and randomness, there is no definite reason as to why this occurred. You know what, guys? I feel a disturbance. I feel like Charles Darwin is upset that we didn't talk about his main Bay mechanism. So, to appease the father of evolution, we might as well talk about natural selection too. Now, as much as I want to look like Gabby Garcia, I can't. There's variation, like hair color and nose shape, and it occurs in all organisms, plants included. Natural selection states that in certain situations, a variation is more favored than others, giving some an advantage in passing their genes. Say for example, the regal peacock. The reason why they fan their tail feathers out is not because they're showing off their beauty to us, but because they're trying to attract a mate. The showier the tail, the higher their chances. For humans like me, it's good that looks aren't the only thing that matters. For the third activity, we started off with another ratio. Number 1. We made our gene pool have an allele frequency of 10/90. Dominant by recessive. Number 2. Next, we selected 50 individuals with replacements and recorded the genotype frequencies without calculating the allele frequencies just yet. Number 3. We were told there was a predator attack that took out half of the light individuals but only 10% of the dark individuals. So we reduced the number of the homozygous recessive by half and the homozygous dominants and heterozygotes by 10% each. Number 4. We calculated the allele frequencies based on this new total. Number 5. After, we adjusted our gene pool frequencies to match what we just calculated, making 1 full generation. Number 6. We repeated this for about 4 generations. Our findings reveal this. In our data, the allelic frequency changed and went toward the direction of the dominance trait because of the predator attack taking out more of the individuals with the homozygous recessive trait and only 10% of the ones with homozygous dominant and heterozygous allele individuals. If the phenotypes were reversed, the same thing would happen, except the more prevalent trait would be light instead of dark. If we started with 100% of homozygous recessive individuals, then no dark individual would appear in the following generations. Natural selection is Darwin's central mechanism driving evolution. But remember that variation is and always will be present. Within our DNA lies a series of 4 letters which are the nucleotides G, A, T, and C, the Great Gatsby. These are arranged in many, many ways and thus give us variation. Fun fact, If you were to string up all the DNA from all the cells of just one person, you could reach the sun from the earth and back 70 times. The way this extremely long string doesn't get tangled up is because it's organized into chromosomes. Each chromosome, plants too, has different amounts of chromosomes, and again, this is because there's variation inside and outside of a species. If two similar species were to interbreed with one another and mix their genetic information, this process is known as the gene flow. If there was a restriction of the gene flow, then each species would only breed within their respective populations. Thus, over time, they will grow more and more different from one another. It's like that one song that goes, "Everything has changed." Activity time. Number 1. We continued using 2 paper bags as gene pools. Number 2. Then we noted initial allele frequencies of each bag based on the results of the natural selection exercise. If they were close to 50 by 50, we exchanged some papers to make them very different. Number 3. For each round, we drew 20 papers or 10 individuals out of the bag without replacements and put them in a small container. Number 4. Next, we swapped the containers and proceeded to dump these papers into our bag. Number 5. We repeated this for about 3 to 4 times. Number 6. Finally, we counted our papers to calculate our new allele frequency. For the last activity, the allelic frequency did also change toward the direction of the dominance trait because during the swapping of alleles, it was simply more present. And we're done! But first, let's do a quick recap on all the topics we touched on. 1. The Hardy-Weinberg equilibrium tells us that allele frequency can remain constant if the population's gene pool meets certain criteria. 2. Genetic drift highlights the randomness of life when it comes to reproduction. 3. Natural selection tells us that some traits are just more favorable than others. 4. Gene flow is where very similar species interbreed. Words I'd like to highlight right now are the words constancy, randomness, favorable, and similar, because it just describes how our lives are. Some of us crave stability and constancy, while others prefer randomness and spontaneity. But one of the things that make us similar is that we just want life to be more favorable. You're not the only one trying to cope, so maybe be kinder to your fellow species. That's it for now. Remember to stay curious, keep adapting, and we'll see you all very soon. Bye!